So I just want to introduce my esteemed panel here. It really needs no introduction. So far as Jason Ehrlich from Genentech, David Boyer from Retina-Vitreous Associates in LA, Charlie Wykoff from Retina Consultants of Houston, and Dante Pieramici. And we've been tasked with kind of looking at the issues surrounding combination therapies in macular degeneration and diabetes. And I'm just going to start with kind of a softball. We'll call it a softball. Dante, is there one of these two diseases that you think is more amenable to combination therapy than the other?
Softball questions are generally good for me.
Yeah.
So thanks for starting with that. But, you know, I think actually both of them are potentially amenable to it. But if I had to pick one, I would say probably diabetes. And I think that's just because it seems like there's much more variability of disease response to our current therapies. And there are many more patients who are left with swelling of the retina or levels of visual acuity that are low that I think that we can benefit with a combination approach. So I would say diabetes, if I had to pick between the two, is more amenable.
Charlie?
Yeah, you know, it's been a bumpy road coming down the combination therapy path, but I think that there is a lot of hope down the horizon, certainly from both a durability and an efficacy perspective from both of these diseases. Just to play devil's advocate, I might take wet AMD because Dante took DME, because, you know, many patients with DME, if you treat them aggressively, you can get the disease to regress, and a lot of patients can have a lot less dosing long term, whereas for AMD, there really is this chronic issue. And we talked this morning about durability issues, but it's not just frequency of injections, right? You're not maxing out the visual acuity curves for AMD, and then you have the atrophy component also. So we need something that addresses both the wet and the dry form simultaneously, and so I think combination ultimately for AMD is the most promising.
You know, I think that dry AMD would be a big hurdle that would really benefit the population. But if I had to pick one for combination therapy, I pick DME, mainly because we leave a lot of fluid in the retina, even with monthly injections. And I think that we all believe that if we can dry those patients out, that we can improve the overall vision of these patients. And you know, Charlie's correct. A number of studies have shown that you give less injections over a period of time. But I still think we leave a lot of vision on the table with DME.
Jason, you're at a company, so how do you guys think about this?
I think we want to have both. No, and whether you could get added efficacy or added durability with the same mechanism in AMD and DME, I think that's yet to be seen. But I think I agree with what everybody's said so far. In AMD, you know, there's two diseases going on, right? You've got the underlying dry AMD component for which there's no therapy currently. So, you know, there it seems like there's a big opportunity to treat the dry AMD along with wet AMD and potentially have better outcomes. In DME, I think the patient population is really quite mixed. There's a lot more components to the pathology that are not necessarily just VEGF driven. So as we start to get better understanding of that, I think there's the possibility of improvements. You know, we and Regeneron both have had some interesting phase 2 proof of concept data in diabetes with you know, multiple mechanisms of action. We'll see how that plays out. But at least it suggests the potential there too.
Well, I mean, so when we look at DME, we understand it a little better. And part of the reasons why I think combination therapy, and I would agree with the people who said DME is a little more likely, is because there are so many other pathways. In AMD, unfortunately, just by the nature of the home run of VEGF inhibitors, it's a high hurdle, and the other pathways don't seem to be working out. And obviously, we'll get into pathways in a moment, but having more pathways makes your disease more amenable to combination therapy. So I want to switch gears because there's a lot of companies in the audience, and I wanted to just kind of touch upon what are the regulatory issues with a combination product. So David, you've run a lot of studies. What are the issues that companies need to be aware of for combination therapy?
I think there's several issues, and they've come up just recently. You know, if you put two drugs that are out there into one syringe, if you don't change it and do what, you know, Genentech did and change the drugs around, you'll end up needing to be superior rather than do a non-inferiority, even though you're giving one injection. So I think you have to look at, you're going to have to formulate these drugs so that you basically change it so that they're a totally different drug. So the other problem I see is a lot of companies come in and say, well, we can give less injections, which would be wonderful for the patients, wonderful for the doctors. But giving less injections still comes down to the bottom line of what's the vision at 12 months or 9 months or wherever the time point is. So even if you gave one injection or if you gave 9 or 12 injections, it really comes down to vision. So I think that the biggest problem is when you do the bispecific, you've now changed the molecule, you now have one drug that's new, so you can be non-inferior. And if you do what Regeneron did and put Eylea, again, with a Tie-2 inhibitor, now you have to be superior. And even though the drug showed a benefit, it showed a benefit in drying, it showed a benefit in length of time between injections, they have to be superior, and I don't think that they're going to be able to reach that bar.
So from a standpoint of superiority, and Charlie, you run also a lot of studies, if you were to sit at an ad board or discuss with a company bringing their combination product, will design a 2As, what type of superiority outcomes would you recommend that they look at? Obviously, at the end of the day, it's all about vision, but there's ways to get things past the FDA, right?
Yeah, no, I agree with everything David said. From a simple clinician's perspective, I'm really not interested in non-inferiority. If I'm going to take it into the clinic for my patients on a day-to-day basis, I truly want something that's better. So if we're in the sandbox talking about how do we get it approved, it's really not superior, it's kind of the same, me too. From a clinician's perspective, simply, I don't need that. I really want the next generation that truly is superior on everything, not just something that's going to get it approved. That's sort of the simple way I see it. I want to win on vision, I want to win on anatomy, and I want on durability. And I think some of the things on the horizon are going to get us there. So I think if we're talking about how can we get it approved, but it's not that much better, I think we should talk about something else.
Dante, does it matter about how easy it is to do the combination therapy? So in other words, our current standard in these diseases is anti-VEGF injections. What happens if you combine that, say, with an eye drop? Shouldn't that have a lower bar?
Well, I think at the end of the day, the thing that matters most is the efficacy and safety of the treatment. So, if one treatment's more difficult than another, I think that really it's going to come down to efficacy. If it's much more efficacious and it's and a safer therapy, then I think that you're still going to choose that, even though the other therapy might be simpler to administrate.
So, Jason, Charlie's correct. At the end of the day, as clinicians, we want drugs and combinations that are better. But stepping back for a second and talking about regulatory, are there any of these superiority outcomes that are easier to hit than any of the others?
Yeah, I think, showing some statistical superiority, it's the numbers game, right? So it may be easier to show statistical differences on OCT than vision. You might need fewer patients. I think the question the regulators will ask is, what does that matter? And it's a question that physicians and patients will ask too, and it's the right question to ask. So, at the end of the day, it's how are the patients' lives improved by the therapy that you're giving them, right? So, in the case of vision, how much better can they see, and for how much longer between treatments? Those are the things that matter to them. So, I think that the regulators tend to be most persuaded by data that are most relevant for patients, and that's generally the bottom line. So, it may be easier or harder in a trial to show imaging or number of injections, but if that doesn't translate into improved vision outcomes, then I think you're looking at non-inferiority studies in vision. And I think that's okay, right? If you look at the evolution of most drug development, let's take statins, I think, are a great example. You started with Mevacor and Pravachol, and then you got Zocor, and then you got Lipitor, and then you had Crestor. And each one of these was an incremental improvement, but if you look over the arc of those, there was a big difference in number of people dying from cardiovascular disease, right? And that's, most drug, you're not usually going from no anti-VEGF to anti-VEGF. That's not generally the history of drug development. So I think incremental improvements that add up over time actually make a difference.
Yeah, and the reason I put this up here, and David already talked about this, is because I can't tell you the number of companies that come to me and say, we're going to do our combination therapy study, phase 2, and we're going to show that we do less injections with our treatments. And if I can convince anything of the people in this room, that's not an approvable outcome. And so it's almost irrelevant to do a phase 2 with a non-approvable outcome. That's great. As clinicians, as patients, we love that. But for you to get a superiority on injections, you'd actually have to prove that you're safer than this monotherapy, which would require thousands of patients. It's just not going to happen. So the one thing I hope all of us are telling you when it comes to regulatory, don't use injections. And as Jason said, OCT is also not approvable, so don't use OCT. The only thing that's approvable is vision. And so that's what you should use, and you can decide what about vision. And going down that route, David, if you're going to tell a company, I got a combination product with an anti-VEGF as the combo plus our product, what would you tell them to use as a comparator? Avastin, Lucentis, Eylea?
For what disease?
You could choose.
Okay. Well, if I choose diabetes, I would say that you have to use one of the branded medications. You know, Eylea seems to be the standard, but I think you can certainly get by with Lucentis in that situation. I think, you know, if you use Avastin and beat it, it's great, but, you know, you have a $50 drug that you're going against, and I think that your data may not be looked at in the proper light. AMD, on the other hand, seems to show, you know, closer efficacy between the drugs. There may be a little bit of a difference between them, but not as great, or not perceived as great. So I think you could probably get by with Avastin, but I think that, you know, if you're really going for a phase 3 trial and you want acceptance by the community, we want to see you go against the best that we have. And I think, you know, to do it in a phase 2 is fine, or phase 1 to see if you're getting a biological effect that's equal to VEGF is fine. But I think when you finally get down to comparing it in a phase 3, that you probably have to go against a branded medication.
So Dante, would you agree AMD, any of the 3 would be fine, DME, Eylea?
Yeah, for the most part, I agree. I think that probably with AMD, you probably want to stick to a branded drug as well. And the reason is, I find, at least in the Avastin that's delivered to me from the compounding pharmacy, there's always a question of how much drug am I actually getting in a syringe. And I think there are definitely people in the community who would not give a non-branded drug to their mother or themselves, and so I think you're going to have some resistance in people using Avastin as a comparator. So certainly for DME, I think most of the studies now are using Eylea as a comparator, because there's a thought in the community that this is probably the strongest drug against DME. May or may not be on a 1 or 2 year basis. It may be similar to Lucentis, but I think Avastin, again, I'd probably stick away from if at all financially possible.
Go ahead, Charlie.
Peter, the comment I would spin on this is, you know, I think that certainly in dry AMD now, we have an approvable endpoint that's not vision. Mm-hmm. And I do believe that we will get there in the DME angle also. We've gotten there with DR also, so, you know, in VMT. So we've certainly seen non-vision endpoints be approvable, and I think we'll get there with early stages of DME. I would encourage companies to continue that discussion, to create data sets that correlate vision with anatomy. There are many studies already looking at that. And yes, there's not a great correlation, but there is a reproducible correlation across the studies that I'm aware of. And I think if we have more data as a community, I think we can convince the regulatory bodies that be in this country and others that is an approvable endpoint. And I hope we get there, because there's a major disconnect. In my clinics, when Ms. Jones is 20/60 today and 20/40 tomorrow, she's exactly the same in her eyes, but the OCT is consistent. Mm-hmm. So OCT is reliable, and I hope we can get there from an approvable endpoint for DME.
Did you have something, Dave?
Yeah, I just want to say I agree with what Charlie's saying. I think that in the future we'll have artificial intelligence be able to look at OCTAs, be able to look at our OCTs and look at our fundus photographs and probably be able to give us an idea of, what the vision is, the visual field and things just based on these. But that's a ways down, and we need to give them a lot of data before they'll ever be able to do it, and it's got to be reproducible. I think there are other visions besides just the ETDRS vision. You've got low luminance vision, which I think is important, and especially when you're dealing with dry AMD, and you've got reading speed. Now, I honestly don't know how the regulatory people view those, but that is another way that a company could find themselves being superior in some aspects.
I agree too with the thought of a biomarker of some type. I think that we've had DME and OCTs and retinal thickness for a long time, and this turns out to be a very poor correlate, I think, with visual acuity and response to therapy. And we're finding out even now more and more that you can leave fluid on the table in AMD and DME and still end up with relatively good results. So it's at least measuring the thickness on an OCT as a biomarker for disease, I think, poor. But I agree with David. With machine learning, we may find things. And this machine learning thing may not be that far off, really. I mean, it's happening very quickly.
Yeah, and I think, whenever it comes to an outcome, at the end of the day, it has to be with vision. And that's what Charlie's pointing out, is right now OCT doesn't correlate, OCT thickness doesn't correlate with vision. So because of that, the FDA won't accept it. They accepted VMT in OCT outcome because that showed that if you got rid of VMT, that correlated theoretically with better vision. GA is an outcome for dry AMD because even though in a dry AMD study, maybe the patient doesn't improve vision, or 20/20, they're extrafoveal GA, you stop the progression, that's going to eventually lead to vision. So Charlie's right. If we can somehow validate an OCT biomarker, say in DME or AMD, then certainly I think that the agency would consider it. It's just right now there's nothing validated. Any other regulatory issues that you guys feel the audience should be aware of in combination therapy?
I just add, I think most people's experience is that the regulators around the world are data-driven, and so I think if you put new data in front of them that helps them change their perspective, they're open to, and they're they ask for that when they're here on these panel discussions. Please bring them new information, right? Mm-hmm. So I think if people are working on novel endpoints, novel methods of trying to understand disease activity, just go talk to the regulators about it. They're very open, and they want to have those discussions. They want there to be new ways of showing outcomes.
You have to validate it.
But they're not going to do it for us. That's our job.
Just to make one other comment, I think that we do a pretty good job of treating the patients when they're in the clinical trials, and their visual results end up doing very well with a single monotherapy anti-VEGF. But if you really look at the data long term, patients are losing vision. They're actually responding poorly to the therapy. So I don't know how you get regulators to realize that this therapy is not something that is sustainable in a long term. We need combination. We need drug delivery, things that are going to make it. Actually, it's a visual improvement response at the end of the day if you can keep people on therapy for a long period of time.
So Charlie, I want to show you these two types of studies. So the bottom is obviously combination therapy, in that the patient is getting 2 injections, drug A, drug B, at the same time. And at the top of the slide is sort of a different thing. And there are some drugs that would work using the top sort of protocol. And the interesting thing is that the regulatory rules of these two, I would call this relatively combination studies, are totally different. What do you say about these two different study designs?
I think it's all mechanism of action, and if it makes sense from a pathophysiologic perspective to treat with drug A and then switch to drug B, and you get a better outcome than some other pattern, then I think it makes a lot of sense, both from a regulatory and from a clinical perspective. And Peter, you've been a leader with this, with Allegro, and I think that data is fascinating, and I hope that we have more molecules in this space that can somehow reset the pathophysiologic clock inside the eye, and then have a stronger response with a drug that we already use. I think it makes sense.
David?
Yeah, no, I agree with Charlie. I think, it's when the study starts. And that's the big thing. If you're, with the new information coming out about diabetes and that we really only drive 40% of the patients, even with monthly injections, that gives us the hope that that other 60%, if we add something to it early in the course or find out if we can identify those 60%, could we change or modify the disease and get better visual acuities, better diabetic retinopathy severity scores, et cetera. So I think if you could clear all the VEGF and suddenly the patient gets a major bolt, if I'm out there in the audience as a company, I don't want to treat against that because those people are gaining large amounts of vision. But if I can find that group of patients that only gains, let's say, 6 letters over a year or loses vision in the other third or 60% of patients, then treating and identifying that group and then starting your drug in that group that is hard to treat, I think it's realistic. I think that study starts, you identify which group of patients you have, and that's what your label will say. Your label will probably say you have to give 1 or 2, and if they fail conventional treatment, then you can use this. But that's what the label would say.
Yeah, and from a company standpoint, understand, where is that red line on these slides? You got to understand where the study starts. You can't take credit for the first injection in a sequential therapy. You can only take credit from where you start your drug or where the combo starts. That's where the clock starts in terms of improvement. So I wanted to go through a few of the drugs that are out there. And I'm sorry if I've missed your drug on any of these lists, but let's just look at the VEGF pathway. And Dante, we'll start with you. Do you think there's any role for combo, and I'm just talking within the VEGF family, so A, B, C, D, and placental growth factor. Do we need a combo in this space, or are our drugs currently good enough?
Well, the drugs that are out there right now really don't hit all the VEGFs out there. They hit VEGF-A primarily, and there's some placental growth factor. But the VEGF-C and D, we're not getting to, and there's some evidence that this may actually even be upregulated when we downregulate A and B. That may be why some patients have persistent disease. So yeah, I do not think we cover them all adequately enough, and I think that there certainly is a role to add in here some other drugs.
And I apologize to Allergan. I should have changed it to yellow from the press release, but we'll get there. All right, so outside the VEGF pathway, because VEGF is really a high bar, let's start looking at some of the other pathways. So Jason, why don't you run us through the thinking at Genentech to look at the Tie2 pathway combined with the VEGF pathway?
Yeah, so I think the rationale for looking at Tie2 or the Ang1 and Ang2 receptors or ligands, it really goes back to work that Tony and Georgiankopoulos did when the angiotensins were discovered, identifying a potential role in vascular stability, on the Ang1 side, promoting vascular stability, and Ang2 promoting instability of the endothelium, and that leads to a series of preclinical experiments suggesting potential roles in diabetic eye disease as well as AMD or neovascular AMD. So, then it's a question of how do you actually construct, a therapy to target both of those at the same time. And, I think that the data so far suggest, at least in diabetes, that you get pretty substantial improvements in diabetic retinopathy severity, which is consistent with that mechanism, and potentially also improvements in vision outcomes in our phase 2. So we'll see what happens when, as people continue to study that pathway. But it certainly seems intriguing so far. Again, it's all sort of grounded in the basic science of when the angiotensins were first discovered.
David, do you think that this pathway combining Tie2 activation with VEGF inhibition would work any better in one of those two diseases, or any disease for that matter?
I think it works better in diabetes than in wet macular degeneration.
So, Charlie, we have heard from press releases and also presentations that the Regeneron Bayer product didn't make it, so quote, failed phase 2. Is that your opinion of the result of that study?
It's hard to know, thinking about the reality, this is phase 2 data that we're interpreting, right? I mean, the overarching hope is, for me, is that, yes, I hope that targeting these other VEGFs that are in phase 2 and Ang2 and VE-PTP are going to be helpful, but we just need robust phase 3 data, and then we'll know a little more. My interpretation of the 2 phase 2 programs together is that there is clearly a biological signal that the addition of modulation of the Tie2 receptor is valuable, and I sincerely hope that it moves forward to phase 3, and I hope that more than one company pursues that. We need to understand this from many angles.
Genentech's data even seems to suggest there may be a superiority in visual efficacy, so that's exciting. I think that's a possibility.
Yeah, I totally agree. Let's move forward. So many companies, and I apologize again to your company if I didn't put you up here, have tyrosine kinase inhibitors with multiple different ways to deliver them. Do you think TKIs alone are enough, or are we going to need to use this as combination therapy? Anybody want to tackle that one? Jason's pulling the mic up.
So I mean, I guess it's, let's see what happens, right? I mean, it's, TKIs, I'd say, are combination therapy. I mean, because, the ones that you've listed up here, at least most of the ones I'm familiar with, are not specific for one receptor. For instance, the ones that are based on some of the oncology medicines, those are pan-receptor tyrosine kinase inhibitors designed for oncology. So I think the question will be, do they adequately inhibit VEGF receptor without causing yet unknown side effects? We don't know yet. I mean, that's the purpose of the experiment, so we'll have to see. But, it certainly is possible that they could even be better, right? I mean, we don't know, because they're hitting multiple receptors. It's going to be a balance of on-target and off-target effects.
And when you're looking at something like a TKI, which is not in the combo realm, but I just have some really good clinical trialists up here, do any of these delivery methods to you seem more appealing for your patients or yourself than any of the others?
Gray bug, I think, is very appealing because it extends the amount of time between injections. Certainly, it's nice to have topical drops, but topical drop delivery is very difficult. Everything works in rabbits because of the large amount of sclera that's visible, and you can get your drug through the sclera. And systemically, X82 seemed to work, but the toxicity in the liver was just too high. So I'm impressed so far that gray bug may have some place in our treatment.
The one thing I would add on your previous slide, I think, was that subcutaneous delivery, I think, is a very attractive route for patients, especially those that are already giving themselves subcutaneous injections of insulin. And so I think if that pans out, I think that would be a huge value from a patient perspective.
Yeah, I mean, when I look at the TKI data, this to me, as a monotherapy to go up against anti-VEGF, it's a super high bar, and I don't know if they're all strong enough, and they're failing when they're trying to go head-to-head. That's why I put it up, are they enough alone? And for some of the companies that are in this space, maybe consider a phase 2 study where you have a combo arm also, because, as we've just mentioned, it may actually work in combination better than alone. So we'll have to see. That's not been proven. So we have about 5 minutes left here. What other pathways should we consider for combination therapy? And this, and I think in particular, would be in DME, because there's a lot of other pathways that I haven't shown here. What pathways would you consider for a combination therapy?
Well, the Integrand Pathway is one that's being looked at already for a number of companies, and I think for diabetes, this seems particularly attractive as a combination target.
And well, most of the companies right now, well, the one company I know of is using the sort of sequential approach. So the idea that integrin antagonists take a little time, so to speak, to achieve their effects. So you need the anti-VEGF to sort of immediately help, and then the integrins catch up and can take over from there. How about the plasma kallikrein pathway?
Yeah, I mean, I think it's hard to know which ones of these are going to really be clinically active. There's really interesting preclinical data for kallikreins. There's really interesting preclinical data for a lot of inflammatory mediators in diabetic eye disease. Which, if any of them, are going to result in better outcomes, we don't know, right? And I don't think the animal models necessarily translate all that well into differential clinical efficacy in people. So, for better or worse, really the best way to figure this out is have a bunch of different targets in the clinic.
How about steroids? So steroids are obviously received approval, at least some of them, as monotherapy, but they actually work pretty good, except for Protocol U, which was designed interestingly, I should say. What do you think about trying steroids with some of these things, or new steroid delivery, I should say, as opposed to what we have?
I think we all realize that steroids are very effective in the treatment of diabetic macular edema. It's the side effect profile that keeps us from using it on a more constant basis. Mainly the fact that the pressure goes up in some patients, and also the fact that they develop cataracts. So if you had a safer steroid, by safer I mean with less side effect in the pressure rise or in the cataract formation, I think you'd have a much higher use of steroids. So whether you put it in a different place, such as Clearside's doing, and if that can reduce the pressure rise, which it may be doing, or possibly reduce the cataract formation, that would be a big step. It's hard to get rid of all the glucocorticoid action and not develop cataracts, though. That's, I think, the biggest problem.
The way I might tilt this is to simply say that, in some ways, DME and DR are public health problems. We're trying to make people better, but if you catch people early and you follow them and you treat them appropriately, this is a very preventable disease, whereas I see AMD very differently. So I really see, ultimately, combination therapy being much more valuable from an AMD perspective, and in that regard, cell death. I mean, we need to go to the core problem in AMD, and I think that we need to understand the basic biology a lot better there, but combination therapy from a cell death perspective is, I think, where the future is from an AMD perspective.
So just in closing, I'll give everybody a chance to kind of give their final conclusion, but say you're meeting with the CEO of a small company and they're really worried about going forward with their product because the only way it'll work is in combination with something else. Dante, what do you tell the CEO?
Well, one of the things I find interesting is that anti-VEGFs have been out for about 12 years now, and I think the first thing I heard from the KOLs was that they're going to be combination therapy 5 years down the road, but that was 12 years ago. And just speaking to the fact that these anti-VEGF agents work so well, the bar is so high, I think it's quite difficult in these cases. So I'm not trying to be completely discouraging to the person, but I do think that really, as you've mentioned before, vision's important, and it's the most important thing at the end of the day for regulatory reasons. But from a clinical standpoint, there's a whole host of other things that anatomy that you can improve, durability that can make things, as long as you can get it through the visual acuity regulation, that I think that there's a lot of other benefits that we need.
Charlie?
I agree with Dante completely. I think we need to not sell ourselves short. Yes, anti-VEGF monotherapy is fantastic, but if you look at the visual acuity outcomes in our phase 3 data, it's actually not that great. We're probably leaving 50 to 70% of potential visual acuity on the table if our ultimate goal is 20/20. So I think combination therapy is the way of the future, and I think we'll get there. I just think we got to find the right targets.
So, David, I'm going to switch the question just slightly for you because I love you. Yeah. So let's jump forward 20 years. And we have 3-4 different pathways that are working, and they're all combined with VEGF. How do we design studies to see if 3 combinations work better than 2 or 1? How do you envision our clinical trials occurring in 20 years?
So in 20 years, to be able to differentiate between the various combinations, because you're going to have different pathways, and you may want A and B, or A and C, or B and C, and you want to try to figure out what's best. The trials may get larger. However, I think that we will have better surrogate endpoints that we'll be able to go to the FDA with. So I'm hoping that we don't have to have 2,000 patients in each arm of the trial. That we'll have some other surrogate way of measuring vision or measuring the visual fields in some of these patients using OCTA and other combinations. But I think that going to your question, it's going to be very important for the companies that have the VEGF at this moment to look in the future because, their patents are running out. And there are going to be a lot of biosimilar companies out there very shortly that would love to combine to show a better. And I think this is a time for people to start looking at combinations that will have to be non-inferior, but to get into the market, but maybe superior in other ways.
Super. Well, I thank my panelists for a very enlightening conversation.